that I post, because the past few of them had said, if you were reading this announcement, reply to this for extra credit. And like only a handful of you have done it. So anyway, that's why I'm doing these announcements in addition to the written announcements. One, because I know you guys don't all read them. And again, the reason I'm doing them early is to save time. But anyway, I got a good email today. So I wanted to share the question with you. The question and the answer, I want to clarify. As far as independent work goes, remember that is now extra credit, but it's still extra credit. So you can get as much as you want. There is no 100-point limit. You can go and go and go and go. Now, granted, you'll get more bang for your buck by fixing your old labs or, watching videos for days that you're absent, things like that. But you can still do independent work. I still highly recommend it. The grading, the way it's graded the way it affects your grade is different now that it's extra credit. It's hard to explain. If anybody wants me to explain it, just meet me online for office hours and I'll explain it. Hopefully I can record it, that meeting, and then share it with your classmates. But anyway, I guess that's it. One last reminder for next week. Next week's lab is another lab that you will do on the internet. But again, unless I tell you otherwise, you need to come in for your lab day and meet in person, right? Bring your computer. You're going to do your online lab in person because people are not doing their lab. So I wanted to remind you of that for next week. And again, if you do it early and I grade it and I look at it and I might say to you, hey, good, you don't need to come in the lab this week. So there's your incentive to get it done early. Anyway, let me start presenting the screen so I can hear you guys, and then I'll ask if you guys have any questions, because right now I cannot hear you. I don't know why it has that glitch, but it does. All right. So first of all, can someone please confirm that you can hear me and see the PowerPoint?
Yes.
Uh-oh. That might be an issue. Let me try this again. Can someone confirm that you can hear me?
Yes.
Yeah.
Okay, awesome. Vance, I can see you said something. Reed, I can see you say something. I can't hear you, but that's okay. As long as you can see and hear me, that's all that matters. Well, that's not all that matters. It would be best if I could hear you too, but that is better than nothing. So here we go. I guess if anybody hasn't, I can't tell if you have any questions. This stinks, sorry. So if you're in person, if you have any questions, let me know. For attendance, we'll do the same thing we usually do. I'll give you attendance words. You email them to me before 9 a.m. Yeah, and that's it. So the first attendance word will be shorts. Like, the things you wear. Weather's starting to warm up. Well, not this weekend, but yeah, shorts. That's your first attendance word. Moving forward. Actually, let's move backwards for a second. So we're talking about tRNA. Actually, let me really back it up really quickly. The first thing I should remind you of is we are talking about translation right now. And before we get into the process of translation, we are talking about the players of translation, is the way your book put it. So we're talking about the parts that are involved in translation before we even talk about the process. So far, what we've talked about is, let me back it up to remind you. All right, we've talked about mRNA. That's the first ingredient. That's not necessarily in this order. This is the order I'm giving it to you. The order doesn't matter. But yeah, the first ingredient we talked about was mRNA, which makes sense that we would talk about that first, because remember, that's what we talked about in transcription. That's the thing. That's the product of transcription. We made the mRNA in transcription. Then we capped it and spliced it, and then it left the nucleus and went into the cytoplasm. And now that's what we have, right? So mRNA is the first. It's your first ingredient. The second thing is the machine that translates the mRNA, because again, we're talking about translation here, and that machine happens to be called ribosomes. And this is really dumb the way I did this PowerPoint, and I need to fix it. But yeah, so we've talked about mRNA. We've talked about the machine, which is called ribosomes. We mentioned the fact that it requires enzymes and energy, right? Specifically ATP. I also put an X through this because that part definitely won't be on the exams, just for your own knowledge. And of course, then we said there's also something called tRNA. Now, this isn't the whole list, but this is what we've talked about so far. And we left off talking about tRNA. So to also remind you before we move forward about the different RNAs we've learned about, again, you've learned about mRNA. The M stands for messenger, right? That is the message. That's what we copied down from the DNA. That's what we got from transcription. The next type of RNA that we are talking about right now, again, is tRNA. The T stands for transfer. So you need to know the difference between those two. You need to know what that is transferring. Why do we call it transfer RNA? What is it transferring? And again, I told you this at the end of the last lecture, but it is transferring the amino acids. So the transfer RNA, where is it at? There we go. Transfers the amino acids. And if you can remember that, then you're good to go. And again, we already talked about this slide on Wednesday, so I'm going to move forward. This is where we left off. I mentioned in that last slide on Wednesday that the tRNA carries the appropriate amino acid and that it brings it to the ribosome because it's reading the code of the mRNA. So really quickly, we're going to show you how that happens, even though I'm not going to ask you about it. So here's a picture of tRNA, not from your book, but this is a good picture. So you can see it's just a regular old strand of nucleotides, right? G, C, G, G, A, U, just strands of nucleotides. That's all it is. In some places, they've kind of hydrogen bonded with each other. So it's looped around. That's what gives it its three-dimensional shape. I'm not going to ask you anything about that. But I did mention that it reads the code of the mRNA, right? And that's where that happens. This says anticodon loop, but your book doesn't get into that much detail. So you don't need to worry about these words, anticodon loop, this variable loop, none of that. Just know that, again, the tRNA, it is the thing that carries the amino acid. And how does it happen? Well, basically, I'm not going to ask you about this. All you really need to know is it's all about structure and function. The structure of the tRNA allows it to happen. And I'll get into a little bit more detail here, coming up, excuse me. So at one end of the tRNA is a triplet of bases, and I already showed you that in this previous picture right here at the bottom, right down here. That is the anticodon loop, or as your book puts it and simplifies it, just the anticodon. The anticodon is complementary to the codon on the mRNA. So let me give you an example. If the DNA strand was, I don't know, C, G, C, if you can read it with those stupid lines, then that means we would transcribe that into a strand of mRNA. Can anybody tell me? Well, shoot, I can't hear you online. Any guesses if that's C, G, C? Any guesses to what this mRNA would be? That's okay. I put you on the spot. It is G, C, G, right? Because G is complementary to C, and C is complementary to G. And you guys do need to know that for the exam. You will get a question like that, except there'll be much more letters. Anyway, so if the mRNA codon is GCG, then the anticodon on this, the thing that's going to recognize that on the tRNA, whoops, is going to be, this is so sloppy. I hate those lines with a passion. Anyway, the tRNA, the anticodon would then be CGC, again, because it's the complementary of what's on the mRNA. And that's how that works, right? So that tRNA, as you're going to see on this next slide, recognizes it, right? There's the anticodon. Both on the left-hand side and the right-hand side, those are showing the same things, just different versions, different ways of looking at it, right? Those are both just the same tRNA, just a different way of looking at it. But yeah, that's the anticodon. That's the part that reads the message. It recognizes the code, the codon on the mRNA and says, hey, that's me. And then on the other end of the tRNA, this is where the amino acid attaches. And to remind you what I told you on Wednesday, every tRNA is specific, right? Everyone has its own codon or anticodon, and its anticodon has a specific amino acid. And that's how that works. Because you already know from Wednesday and Monday that there's a code that says, bring me this amino acid, right? Each code, each three-letter code says, bring me this amino acid, bring me that amino acid, right? There's 20 amino acids, and there's a code for each one of them. Actually, there's more than one code for each one of them. But any questions so far? All right. So that's it for the tRNA. That was a lot of information. Again, I would say mostly what you need to know for the exam is that the tRNA is the thing that brings the amino acids to the ribosomes, right? It's the thing that carries the amino acids. Maybe I'll ask you some of those other details, like, well, how does it recognize it, right? You know, I might ask about the codon, the anticodon, or I might ask about the fact that there's a specific amino acid for each tRNA, but most likely not. Most likely you just need to know it's the thing that carries the amino acids. And again, to remind you, hopefully I don't need to remind you this, but I keep saying it brings the amino acid, brings the amino acid. Hopefully in your brain you know what I'm talking about. But to remind you, what we're talking about is building proteins, right? And a protein is nothing but a string of amino acids. So that's why I keep saying bring the amino acids, right? Those are the building blocks. Those are the monomers. So the tRNA is the thing. Like if I'm building something, let's say I was building a brick, I don't know, grill, and I needed different bricks, and I had different people. I would say, hey, okay, I need a 12-inch brick. And I would holler it out, and somebody would say, okay, and they'd come and bring me a 12-inch brick. And I'd say, okay, I need an 18-inch brick. And I'd holler it out, and a different person would come and bring me an 18-inch brick. And I keep doing that until I built my brick grill, right? So the people bringing me those bricks, those would be the tRNA. And the bricks themselves, those would be the amino acids, right? Anyway, ribosomes. This is the next part. So again, we're talking about the parts of translation. We haven't even talked about the process yet. We're talking about the parts. We've talked about mRNA. We've talked about tRNA. I've mentioned ribosomes, but now we're actually going to talk about what they are. First of all, they are organelles. I'm not going to ask you about that. You don't need to know that they're organelles. That's just for your own knowledge. What do these organelles do? They coordinate the functioning of mRNA and tRNA. So again, as you already know, because we just talked about it, mRNA, that's the messenger, right? It's the thing that has all the codes that says, bring me this amino acid, bring me that amino acid. And then the tRNA is the thing that says, okay, I'll bring you this amino acid. I'll bring you that amino acid. So the ribosomes are the things that bring these two parts together. Therefore, if you think about it, this is the machine, if you will, that makes the polypeptides, which is what we're after, right? Because translation, the purpose of translation is to make polypeptides / proteins. So for the sake of this conversation, those two words are interchangeable, because remember, a polypeptide is a string of amino acids, and once it folds into its proper shape, we then consider it a protein. But again, for the purposes of this particular conversation, those two words are interchangeable. So we're trying to make proteins, right? That's what we do in translation. And the ribosome is the thing that brings together the mRNA and the tRNA. And obviously, if it's a tRNA, it also has its amino acid attached to it. So really, that's all the parts. But let's keep talking about how this works. Technically, the ribosome is not one piece. It is two subunits. Each of those subunits are made up of proteins and yet another kind of RNA. So let's make things a little bit more complicated. Now this is the third type of RNA you need to know about. So we've talked about mRNA, we've talked about tRNA, and now you have to know rRNA. But this one's the easiest because it's in the it's in the name, right? Ribosomal RNA. Yeah, that's the thing that makes up ribosomes. Now for the exam, I don't necessarily need you to know that it's made up of two subunits. I don't really care about that. It's not very important as far as understanding the big picture of how translation works. So that's why I won't ask you about it. I might ask you might need to know that a ribosome is made up of protein and ribosomal RNA. I'm not sure. But at the very least, if I ask you about ribosomal RNA, well, of course, that would give it away if I ask it that way. At the very least, if I ask you what rRNA is, you need to think, oh, ribosomal RNA. That's the thing that makes up the ribosome. Anyway, as this bullet point right here says, it coordinates the functioning of the mRNA and tRNA. Therefore, it should make sense that ribosomes have a binding site for the mRNA and the tRNA. And yes, technically, the mRNA binds on the small subunit. And yes, technically, the tRNA binds on the large subunit. That'll make more sense when I show you the picture. But I'm putting an X through it because I'm not going to ask you that picture. Or I'm not going to ask you that question. You don't need to know those details. Matter of fact, I guess I could just put an X through this. Because this bullet point here is something you do need to know. It coordinates the functioning of the mRNA and the tRNA. So, of course, if you know that, then, of course, it's going to have a binding site for those two things. That's just extra information. I would ask you if there's any questions, but I can't. Well, I'll try anyway. Yeah, are there any questions? And it's just silent. I hope none of you have questions. If you do,
No.
it's funny. It's almost ironic. If you read my message yesterday, which, judging by the fact that no one replied back for extra credit, I assume none of you did. But
I replied.
in my announcement yesterday, I reminded you guys that this is an in-person class, not an online class, and I'm doing the best I can to record these. But remember, it is an in-person class. So if something doesn't work out for you, like it is today with the audio, I apologize, but
he really can't hear.
Not my fault. It's not your fault. It's just the way it is. That's the risk you run by not coming to class. Anyway, here's a picture. Here's everything we're saying. Yeah, here's the ribosome. Again, like I said, it's made up of a large subunit and a small subunit, but that doesn't matter. I don't care that you know that. It has an mRNA binding site. It has a tRNA binding site. I don't care that you know that. Your book even gets specific and says there's an A site and a P site, and we'll talk about the A site and the P site, but I'm not going to ask you any details about the A site and the P site. Just know that the tRNA has a place to go. And as a matter of fact, your book is even giving you the easy version because there's actually 3 binding sites. So I don't even know why your book would teach this because it's inaccurate. But anyway, basically what happens, and if you're taking notes, give your hand a break because we're going to talk about this in detail. So this is just an overview. But now that we've talked about all the parts, here they are, right? The blue part, that's the ribosome. Here's the red mRNA. That's the product of transcription, right? That's what we made when we copied the DNA. We have that mRNA. And then here is the tRNA that's bringing in the amino acids, right? All the parts. They're all right there in that one picture. Now, when we move forward, we're going to figure out how all this happens. For the person for you in class, do you have any questions so far? Okay. Ooh. If it's not too much, and you don't have to do this if you don't want, I'm not even going to ask. I'm not going to follow up. But if you could get online, and then that way, if somebody has a question, if they type it, maybe you'll see it and you can tell me. But you don't have to, and you don't let me know if you do or not. But that would be helpful.
Hello.
Okay. We are now done talking about translation. We are now done talking about the parts, the players of translation. Now let's talk about the process. And really quickly, I've got to check something.
Hello.
Give you guys time to let everything soak in.
Can you hear me?
Okay. Anyway, the next word for attendance is T-shirt. Like something you would wear. T-shirt. So here's some good news. The same translation is also broken down into 3 stages, just like transcription was. And to remind you, the 3 stages, the 3 phases of transcription in order, what you needed to know in order, were initiation, elongation, and termination. Yay! Very rarely do things go easy in biology or they have the same name. So there you go. Same names, initiation, elongation, and termination. So you do need to know that for the exam. You do need to know that transcription is broken down into initiation, elongation, termination. You do need to know those in that order. Same thing for transcription. You need to know it's initiation, elongation, termination, and you need to know that it's in that order. Your book goes into deep detail on this, and so will I because I teach out of the book. But pay attention because I'm going to tell you what you need to know for the exam and what you don't. That's another reason I'm going over what your book says, because I want to be able to say this you need to know and this you don't. So yeah, the same 3 phases as transcription, at least by the name. Now it works differently, obviously, because we're talking about translation and not transcription, but at least they have the same name. Whoops. I can't hear you guys anyway. So what I was going to do was ask you before I get started was, what do you think initiation is? Obviously, that's the beginning of translation, but I was going to ask you, what do you think happens in translation? And hopefully, if you sat and thought about it for 15 seconds, common sense would tell you, right? What we just talked about prior to the process of translation was the players of translation. So hopefully, common sense and logic would tell you the first step must be bringing all the stuff together. All that stuff we talked about, well, it needs to be together for this to happen. So that must logically be the first step in translation. And it is. That's when we bring together the mRNA, which, by the way, has the cap and the tail because it's been modified before it left the nucleus. And then your book gets specific. Yes, it brings the mRNA, and then it binds to the ribosome with the help of the cap and tail. But guess what? Big ol' X through that. Because again, I want you to know the big picture. Now, don't get me wrong. If you can memorize all these little details, then you have a better chance of memorizing the big picture. But I just want you to know what you're going to be responsible for.
Big and black and white.
And a picture of this is going to happen on the. I'm going to show you a picture of this on the next slide. So yeah, it's bringing together the mRNA with the ribosome and the other part that we talked about, which is the tRNA and the amino acid. So let's look at a picture of it. Well, there's another picture of mRNA, right? That's the product of transcription. We transcribe the DNA. We're left with mRNA. We splice all the junk out. We add a cap. We add a tail. It leaves the nucleus. It comes into the cytoplasm. And here we go in translation. So technically what happens, and it happens in this order. Technically, remember, I said you didn't know this, but still remember that the ribosome is technically two parts. It's the small subunit and the large subunit. And they're not together. It's not like they hang out together. So the small subunit is where the mRNA attached. And I already told you that, but I already told you didn't need to know that, but I'm teaching it. So what happens is the mRNA attaches, and then that first tRNA comes in, but yep, I'm the first one. And by the way, it always has this amino acid, MET. You don't need to know that, but there it is. So anyway, the mRNA comes in, it attaches to the small ribosomal subunit. Next thing comes in the tRNA with that particular amino acid. And then finally, the large subunit comes in, and now all the parts are together, right? But for the exam, let me look at this closely. Yes, put a big old X through this. Yes, technically, the mRNA binds to the small subunit before the large subunit comes in. And yes, technically, that first amino acid comes in before the large subunit. But I don't care about that. All you need to know is that all the parts come together for initiation. What is initiation of translation? It's when all the parts come together. What are the parts? The ribosome, which is this brown thing. It's a picture not from your book. The ribosome, the mRNA, and of course the tRNA carrying its amino acid. That's it. Any questions? All right. So this should look familiar. This is the same process, except this picture is from your book, unlike the last one. Same thing, though. mRNA attaches to the small subunit. The tRNA comes in with its amino acid. Then the large subunit comes in, and everything's ready to go. But again, you don't necessarily need, or you definitely don't need to know that in that order. Of course, now that I'm thinking about it, that might be a good extra credit question. So I might have extra questions where I'm like, all right, you know the three parts of translation. Now tell me what happens first in initiation, right? I might ask you that for extra credit, but you certainly don't need to know it for regular credit, so to speak. Anyway, moving forward. If you download the PowerPoint, you can watch this animation that shows translation in action. But honestly, some of the best videos that are out there, I'm going to share them with you. I shared one yesterday. That one of those quick biology crash courses. I highly recommend watching it. Even if you don't do it for extra credit, just watch the video. It'll help you study. All right. We are now talking about the second part of translation, which is elongation. So we're done talking about the first part, which is initiation. We're now talking about elongation. Elongation is when the amino acids are added one by one to the first amino acid. Each addition occurs in a three-step elongation process. Let's stop talking before we move forward. First of all, let's look at how this is the same as transcription. How is this similar to transcription? So remember, elongation in termination and transcription. What were we elongating in transcription? We were elongating the mRNA strand. Of course, this is translation, so we're not making mRNA. We're using it to make a polypeptide. How do you make a polypeptide? You keep adding amino acids, right? So it would make sense, hopefully logically this makes sense to you, that the next stage, elongation, what are we elongating? We're elongating the polypeptide. So logically, it should make sense that elongation is simply adding amino acids. That's what we're doing. In shorthand, that's what you should know for the exam. What is elongation of translation? It's adding amino acids to the polypeptide. Amino acids added, I guess, if you want to shorthand it. That's it. Of course, and again, I teach out of the book, including my PowerPoint. So there's a sentence on your book that says the amino acids are added one by one to the first amino acid. But now that I'm thinking about it, if I ever edit this PowerPoint, I'll probably just get rid of that. Of course, it's added to the first amino acid. Well, you're not going to add amino acids to the last amino acid because the last amino acid's not there until the end, right? So yeah, again, elongation, what are we doing? We're adding amino acids. Now, here's where it gets a little bit more complicated. Each addition occurs in a 3-step process. I'm going to teach you this 3-step process, but I am not going to ask you about the 3-step process. Of course, now that I'm thinking about it, sure, I might give you some extra credit questions for it. But for the exam, what you need to know about elongation of translation is that is when we are adding amino acids to each other. We're building the polypeptide. We are elongating the polypeptide. So yeah, let's talk about it. Here we go. Whoops. Yeah, first step. Sorry, the first step is right here on the page. Step 1, and this should be very logical to you. If we had time to really talk about it and I were to ask you about it, hopefully you could recognize this and come up with this on your own. But obviously the first step is going to be codon recognition. Right? If we're bringing in amino acids, we can't just do it randomly, right? We need to have the right amino acids. So obviously the first step must be that the tRNA that's carrying its specific amino acid needs to recognize its code on the mRNA, right? So there's a particular codon right here that's not shown. Like we don't know what the letters are in this picture. Oh, there we go. Damn those lines. Anyway, right there is the codon. Right? So the tRNA recognizes it because remember, there's a tRNA for every, well, there's more than one, but there's a tRNA for every amino acid and there's 20 amino acids. So we need to know which one, right? And those things are just floating around in your cytoplasm and it recognizes it. The correct tRNA says, oh, that's my codon. This is me. Let me bring my amino acid because that's my codon. And it comes in right here and that's what happens, right? So step 1 is obviously codon recognition. So try to picture this in your brain. Of course, you're the only one I can talk to in person, but try to picture this in your brain. This is step 1. Now imagine this actually happening. Imagine if I could grab this thing and put it right there. And now this thing is sitting right there. Here we have this chain of amino acids. And then we have this amino acid sitting right next to that chain of amino acids. Any guesses to what step 2 might be? Knowing that this thing is here. And we have a chain of polypeptides here, a chain of amino acids. And this amino acid is just sitting right next to that chain of amino acids. Any guesses what step 2 might be? That's okay. I have a feeling if we weren't in a rush and I could actually give you some time, you might come up with it, but I'm rushing you and putting you on the spot, and you're the only one here, so don't feel bad for not getting it. But hopefully this would be logical to you guys. I wish we had more time. If you're watching the video, pause it and think about this. What must be the next step? What we're trying to build is a polypeptide, which is a string of amino acids. We're making it longer, longer, and longer. And what happens in this first step is this thing's coming right here, right? This amino acid is going to be right next to this chain of amino acids. And this chain of amino acids is what we are trying to elongate. So therefore, and again, pause it if you're watching the video, try to come up with it. But here we go. Here's the answer to what happens next. And this should be logical. Obviously, since we're building a chain of amino acids, that amino acid that was right next to the chain of amino acids, that's going to be added to the chain of amino acids. That's truly the elongation part, right? Here's the part where we're actually truly elongating because that amino acid that was all by itself is now going to be attached to the chain. Your book gets pretty specific and shows that the chain technically goes to the amino acid as opposed to the amino acid going to the chain, but that doesn't matter. The point here is in step 2, we're truly doing the elongation. Step 2 is when the amino acid is being added to that chain of polypeptides, right? And again, your book gets pretty specific. First of all, I'm not even going to ask you about step 1, step 2, and step 3, except maybe an extra credit. But even if I do ask you that in extra credit, I'm certainly not going to ask about the A site and the P site. You don't need to know. You definitely don't need to know that. Again, if anything, just know that step 2, that's when we add the amino acid to the growing chain. The chain has one more amino acid. That's why I put that in bold. If anything, you just need to remember that. So now try to picture this. And sorry, I'm going to put you on the spot again. And again, don't feel bad if you don't get it. Because I'm putting you on the spot and you're the only one here. And I'm not going to give you much time. But what's happening, try to picture this in your mind. So it shows that this thing's going to be attached, right? So now imagine that this thing has the chain of amino acids. And this thing has nothing, right? It doesn't even have an amino acid anymore, much less a chain of amino acids. So this thing has a chain of amino acids, which is what we're growing, a polypeptide. And this thing is now basically useless. So any guesses what the next step might be? Also, let me give you another hint. Even though I told you we weren't talking about the A site and the P site, at this point, both of the sites are filled up. So we know we need another amino acid eventually, but that has nowhere to go. So knowing that this thing's useless, this thing has the new amino acid chain, and we need somewhere for the new tRNA to go. Any guesses to what the next step might be? What's that? Nope, that's okay. And again, if you're watching the video, pause it and think about it, and maybe you can get it. Hopefully, logically, this has to make sense. We need another tRNA to come in for once we go through another round of elongation. That tRNA, the new one with a new amino acid, has nowhere to go. So what do? We kick out the old one, right? We don't need this one anymore. It's useless. It did its job. It gave us an amino acid. It's gone, right? That thing moves away. Now this tRNA that has the ever-growing chain of polypeptides, it moves over and takes its place, right? That's basically it, right? And then that brings in the next tRNA codon, which will be the next step, right? So step 3 is called translocation. That's just when we're kicking out the useless tRNA, right? Get out of here. We don't need you anymore. That's it. So step 1, all the parts come together. Step 2, you actually grow the amino acid chain. Step 3, we get rid of the useless parts so you can open it up again and start the process over again. And there it is, all in one picture. So again, for the exam, you don't need to know step 1, step 2, and step 3 of elongation, but I might have extra credit questions for it. And that's it. Any questions? All right, I'm going to let that picture sit for a second and let you guys think about that. All right. And the next question, the next word for attendance is chicken, like the thing that you eat or the thing that crosses the road in a joke. All right. So here we are, finally, to the third and final stage or phase or step of translation, which is called termination. So keep in mind, again, just like we talked about with elongation, let's also look at the similarities between this and transcription. So remember, in transcription, the final phase is also called termination. And in transcription, I even asked you, where is this? Where does this happen? Right? Where on the DNA does termination happen? And if we're talking about DNA, which means we're talking about transcription from earlier in the week, that happens at the terminator, right? So the equivalent of that, where does that happen for translation? Where on the RNA does that happen? It's called the stop codon. So the stop codon is where it happens, and you need to know it. Just like you need to know for transcription, it stops at the terminator. Well, for translation, it stops at the stop codon. Ah, man, and that reminds me. Of course, this should be very easy to remember. The slide didn't say this, but when we talked about initiation of translation, I didn't tell you where it starts, but it starts at the start codon. So translation is the easy one. It's in the name, start codon and stop codon. Transcription is a little bit different because it's called a promoter and a terminator. But here, it's literally in the name. It's the stop codon and the start codon. So you do need to know that. What you don't need to know is what those codons are. There's 3 of them. So UAA tells all the parts we're done. So does UAG. So does UGA. You don't need to know that, but there you go. Those are the stop codons. Those are the codes that tell the machines, if you will, all right, we're done. Our job here is done. It does not code for an amino acid. It just says stop translation. Put an X through that. It's true, but I'm not going to ask you about that on the exam. So again, if I had more time, I would ask you, what do you think happens in termination? And hopefully it would make sense to you that it's basically the opposite of initiation. Because in initiation, we bring all the parts together. Well, guess what? For termination, all the parts basically break up. That's it. The parts go away. That's all you need to know for the exam. This picture is not from your book. But there it is, right? Here we have all the parts. There's something called release factor. Again, this picture is not from your book, so you don't need to worry about that detail. There's a stop codon. That tells everything. You're all right, we're done. So as Fleetwood Mac says, you can go your own way, right? So now all the parts go their own way, and we are done. Now, there's a question on the study guide that might be on your exam. And the question is, what would happen if you had a mutation that caused a change in the stop codon to no longer be a stop codon? What would happen if there was a mutation and then there was no stop codon? What would you end up with? And if we had time, I would, and if I could hear you guys online, because I'm tired of putting you on the spot, that's not right. Hopefully you guys could think about that and come up with this answer. It makes sense. If you use no stop codon, then you would come up with an extremely long mRNA, right? Or excuse me, extremely long polypeptide, because if there's nothing telling it to stop, it's going to keep going. Or what's more likely to happen is some other code is going to have a mutation that does a premature stop codon. So rather than having no stop codon, let's say, let's see. Here's something that's not up here. Let me write it down. Now, I don't know this off the top of my head, but UCC, I know that's not a stop codon. I know that off the top of my head. So UCC codes for an amino acid, right? Let me pick a different one. Sorry, guys. I wasn't thinking. I've got Friday brain. I'm looking forward to the weekend. How about this? I hate those lines. UCA. All I know about that is that is not a stop codon. And I used to know what it coded for. If it codes for an amino acid, I used to know which one. I don't now. Now, UCA is nothing, right? Or it's not a stop codon. But let's say there was a mutation and that C turned into a G. Or that C turned into an A. Then all of a sudden, what used to say, hey, bring me this particular amino acid, now says, stop, we're done. And that comes back to the question that's on the study guide that might be on the exam. What would happen if you had a premature stop codon? And I wish I had time to ask you this, because if you missed this question right now that I'm asking, then you're not paying attention. Because I just told you if there's no stop codon, you would have an extra long polypeptide. So now I'm asking you the opposite. What if there's a premature stop codon? Well, your polypeptide would just be too short. So there we go. Know that for the exam. Any questions? All right. And that's important too. We're going to talk about mutations. Mutations are very important. Woo! Moving forward. Now that we've talked about translation and all the players and the processes, and we've also talked about transcription, which is going from DNA to RNA, and obviously translation, which is RNA to protein. Now let's review it very quickly. Now that you have all that information, let's again just scratch the surface and not go into details, but jog your memory and put all this stuff, all this information together. So the flow of information, again, going from DNA to RNA to protein. First of all, like I keep saying, that's how I recommend studying it. Study the big picture and then add details. So that's the first big picture. We're going from DNA to RNA to protein. Once you have that memorized, then remember, okay, like I said, we're going from DNA to RNA. Oh, yeah, that has a name. That's called transcription. Okay, and then we go from RNA to protein. Oh, yeah, that has a name. That's called translation. Then no, transcription, like I mentioned in this last lecture, transcription happens in the nucleus, and it makes, obviously, RNA, because it goes from DNA to RNA. That RNA has to be processed before it leaves the nucleus. We know all that. Then it leaves the nucleus, and that's where it is then translated into protein, right? What does it? What does the translating? Yeah, it's the ribosome that does it. Let me put an X through this. because you don't need to know it. I'll just put a line through it. Yes, one ribosome makes an average size polypeptide in less than a minute, meaning it's very quick. But I'm not going to ask you about that. It's also not technically a review because I didn't include that on my PowerPoint earlier. But anyway, that's translation. Now again, I'm not going to ask you about this either, I don't think, maybe for extra credit. But remember, a polypeptide is nothing but a chain of amino acids, right? Just like starch is nothing but a chain of glucoses, or a pearl necklace is nothing but a chain of pearls, or a train is nothing but a bunch of trains linked together. Well, a polypeptide is just a bunch of amino acids put together. And it's not yet a protein until it folds into its final shape. Just like a piece of paper is just a piece of paper until you fold it into a paper airplane, at which point, yes, technically it's still a piece of paper, but more importantly, it is a paper airplane because it is folded into its proper final shape. And without that shape, it's nothing, right? A paper airplane will not fly unless it has the proper shape. Just like a protein, it has to have its proper shape to do its job. Whew. All right. Here's a picture of everything we said. There's your DNA that has the gene inside the nucleus. The RNA polymerase, you need to know the name of that thing. It attaches to the DNA. It opens it up so it can read the code. It uses that code to build the mRNA. That mRNA is done, but we get rid of all the junk mRNA, which is called the introns. But we keep the good stuff, which is called the exons. We also add a cap to it. We add a tail to it so it doesn't get destroyed when it leaves the nucleus, and also because we need the ribosomes to recognize it. So it is not ready to leave the nucleus. It does, all right? And then all the parts come together. And we have enzymes. We have energy. Oh, yeah. I'm going to put it next to this. We didn't even talk about this. But, yeah, I keep saying that the tRNA carries its own particular amino acid. I didn't even talk about the fact that, obviously, that amino acid has to make it onto the tRNA. But, yeah, we don't even worry about that detail. Anyway, here we go. The mRNA leaves the nucleus. This is step one. This is of translation, right? This is initiation of translation, which is where all the parts come together. The mRNA, the ribosome, the tRNA carrying its amino acid. What comes next? Next, obviously, we're going to elongate the polypeptide. So we're going to keep adding amino acid one by one. Finally, we hit the we hit the stop codon because we're at termination. And that tells everything to just break apart. Everything goes its own way. And that's the end of it. Whew. So, let me just really quickly read through this. Transcription and translation is how genes control the structures and activities of cells. We already pretty much said that. We're just, again, this is a review. So this is how we control the structures and activities of cells. The structures make sense, right? Because I keep saying genes are instructions on how to build a protein. So it's all about build this, build this, build this. I keep saying this. But I also want to take this time to remind you, a lot of the times, those proteins are just enzymes, right? And enzymes are things that catalyze chemical reactions. Those are the things where, like, without those enzymes, those chemical reactions wouldn't happen. So that's what we mean by activities, right? So without these proteins, those particular chemical reactions just wouldn't happen. Especially the ones you've learned about. You've learned a lot of chemical reactions in photosynthesis and respiration. We didn't talk about them in detail, but you know there's a lot of them. And this is why, because there's proteins doing them. There's enzymes in charge of them. Anyway, as we already know, the information originates with a specific sequence of nucleotides in the DNA gene, the A's, the T's, the C's, and the G's. This is a review. I've already told you this, which is why I'm going through it so quickly. That particular gene dictates the transcription of a complementary sequence of nucleotides in the mRNA, which is a really long-winded way of saying, if the RNA code, if the DNA code is CCT, then the complementary code, so when you make the RNA, it's going to be GGA. And again, you do need to know that. The mRNA sequence specifies the sequence of amino acid sequences in the polypeptide, which is a long-winded way of saying this sequence, this GGA of the on the RNA, that's a code that says bring me this amino acid or that amino acid, right? It says bring me a particular amino acid, one of the 20. It says bring me that amino acid. That's what this bullet point right here says. And then finally, the proteins from those polypeptides determine the appearance and the capabilities of the cell and the organism. So these two bullet points kind of go together. But anyway, any questions about the review? I know this is a complicated thing, so please, if you have questions, visit me during office hours. Let me, if you want me to explain it slower or focus in on one particular part of this that I've talked about, let me
Can
know.
you hear me?
And remember, you do get extra credit, like I said at the beginning of the semester. You get extra credit just for visiting me during office hours. Now, mind you, you have to remind me that you have to ask for the extra credit. But anyway, so again, the DNA to RNA to protein pathway. I should say again, this is new information. This used to be, believed that it was the only way that genetic information controls traits. We used to think that was it. But there have been recent discoveries that points to more complex rules for RNA. So far you've learned about mRNA, messenger RNA. You've learned about tRNA, which is the transfer RNA because it's transferring amino acids. You've learned about rRNA, which is ribosomal RNA. It's the things that makes up the ribosomes. But there's so many more RNAs out there. It is really quite. So there's a lot of ways and sort of, how do I put this without getting too confusing or too incorrect? Some of the RNA that's transcribed, it turns into things that act a lot like enzymes. So they go out in your cell and they do work themselves. Like you don't even need to translate them into proteins to do work. Sometimes those RNAs themselves do work. And again, it's chapter 11 in your textbook, but we're going to skip it. As a matter of fact, we're also going to skip chapter 12 and jump straight into chapter 13. All right, really quickly. Let's talk about mutations. I've already hinted at this. Now, this is a very important bullet point for the entire semester. Everything you're learning in this chapter is going to be important to understand the upcoming chapters, especially this bullet point. Even though it's a very simple bullet point, there's not going to be many questions about it on the exam. This bullet point has a lot of implications for the next chapter, which is about natural selection. Oh, here's a good time. I'm going to say this really quickly. Very quickly, let's talk about these moths. There was a bunch of light-colored moths, right? And then by freak accident, there happened to be a mutation. In one of those white-colored moths, it had offspring, and those offspring came out black, right? That had nothing to do with a choice. That white moth wasn't like, oh, man, it's getting dark out here. I better pop out some dark offspring. No, it was completely random. It was a mutation. No decision was made. That was not any kind of thing for survival. It just happened to happen. And then it just so happens that when that happened, when there happened to be this brood of a few dark-colored moths from that one light-colored moth, it just so happened that it was dark in the surroundings, right? So it just so happened that that mutation at that point was advantageous because now those dark moths could really hide. They didn't choose that. They were just born that way. So now they were able to hide from the predators better than those light-colored moths. So over time, those predators kept eating the light-colored moths because they couldn't see the dark-colored moths. So eventually, over time, your population went from being mostly a bunch of light-colored moths and a few dark-colored moths to being mostly a bunch of dark-colored moths and very few light-colored moths. You understand? I hope you understand that. None of that was on purpose. No single moth ever changed colors. No moth decided, oh, I need to hide. None of that happened. It was all random. Anyway, and that was all because of a mutation. And mutations are random. Now, let's move forward. Actually, let me let you think about that for a second while I check. Oh, come on. Man, you're killing me. What? How did I hit that button? Good grief. I'm sorry about this, guys. Here we are behind on time, and I've got to go back and back and back and back and back and back. Anyway, while I'm doing this, the next word for attendance is going to be toast. You know, like the thing you would eat, like bread that you slightly burn until it gets crusty. Toast. You son of a gun. I'm really irritated that this happened. Hey, here we go. Come on. Mutations almost there. Really want to finish this chapter by Monday. If we finish the chapter by Monday, we can either have the exam on Friday or Wednesday or Friday. If you have an idea of what you would prefer, send me an email and let me know. So far, one of you has sent me an email, and that student prefers to have the exam a day, prefers to have a day between when we finish chapter 10 and when we have the exam. So anyway, mutations, finally, thank goodness. A mutation is any change in the nucleotide sequence of the DNA. So here's a good example. Here's an original sequence: TAA, CTG, CA, GGT. That's the original. Oh, but look, we changed one of them. We changed that T to a C. That is a mutation. It's completely random. I'll remind you back when we talked about DNA replication, I said that there's a machine that does it, and you need to know the name of it. That is called DNA polymerase. The DNA polymerase reads the original code of DNA, and it makes a new strand of it based on the code of the original strand. So I said that it's very fast and very accurate. I didn't say it's perfect. So sometimes it does make mistakes, and that's what a mutation is. When it makes those mistakes, then you get the wrong letter. Your book does point out, even though I'm not going to necessarily ask you this, this is for your own knowledge, that these mistakes can involve large regions of chromosomes. So, for example, too, you could have a mistake with the crossing over. Remember that from chapter 8 when the homologous chromosomes come together, and then you end up with a mom chromosome with a little chunk of dad, and a dad chromosome with a little chunk of mom. Well, sometimes that goes wrong, and you have large regions of chromosomes that are completely messed up. Sometimes, like in this picture, it can just be a single nucleotide pair. So here, again, we changed a T to a C because that DNA polymerase just messed up. Or it can be anywhere in between, right? So it can be large, it can be small, it can be anywhere in between. Again, I'm not going to ask you about that. That's just something for your own knowledge. Here's a picture that's actually from your book because it uses sickle cell as an example, which is great because we're going to talk about sickle cell again later in the semester. So keep in mind, try not to forget about sickle cell. This is not a hypothetical example like I like to give. This is a real life example. So you have a gene that codes for hemoglobin. Hemoglobin is the protein in your red blood cells that basically grabs the oxygen, says, all right, thanks, I'm going to keep that, right? So without the hemoglobin, you don't have oxygen, and you're dead. So anyway, you have a gene. There's a particular codon, remember the three-letter words, that goes CTT. That's what it's supposed to be. But for people who have sickle cell syndrome, or sickle cell, there's a mutation. So instead of CTT, they have CAT. And of course, if you have the wrong DNA code, then when transcription happens, you end up with the wrong mRNA, right? So now instead of having the GAA like you should, you have a GUA. And big deal, you say, right? What's the big deal? It's one letter. Well, in this case, remember, these words say, bring me this amino acid, bring me that amino acid. So this particular word, GAA, says, bring me this amino acid. Glutamine, I think. Glue. We'll call it glue. Now this one, all we did was change one word or one letter in the word, but that now says bring me a completely different amino acid, valine. So now because we changed one word, one letter of one word of the entire gene, we now have a different amino acid. But remember from chapter 3, when we talked about amino acids, they have those different side chains. Some of them are hydrophilic, some of them are hydrophobic, some of them are basic, some of them are acidic. Some of them are charged, some of them are not charged, right? So they have different chemistries. So because of that one wrong amino acid, what happens is it has a completely different shape. And remember, shape is important for a protein. So if you mess up the shape, you mess up the protein's function. So that hemoglobin just doesn't work. And we'll talk about that again later. But anyway, that was just an example of a mutation. Of course, it's 8:51. We're done. Sorry, guys. Have a good weekend. Sorry I kept you over. Send me your emails. The last word for attendance is going to be tint. There we go. I'll be online for office hours if you need me. It's 2 days in a row I've gone over. I apologize. I'll give everybody a little bit of extra credit who was on time and you. Oh yeah, can you please send me an email just to remind me that you're here and so I can have a paper trail?